Padrid, and this is my cell signaling project. My project is on ropanol, which can also be referred to as roofies. Ropanol is classified as a drug known as benzodiazepine and has gained a lot of attention as it is approved and manufactured outside of the US, although that is not the case for inside the US as it is illegal. It was originally created for patients who needed deep sleep, but sadly found notoriety for being the date rape drug. My drug correlates with what is called GABA or gamma aminobutyric acid, which plays a role in the central nervous system. It occurs in every organism that has a nervous system and is the ligand that has two receptors. I will also show how it normally functions first without rohypnol and with rohypnol. It acts as a major inhibitory neurotransmitter. What that means is that when GABA reaches and binds with a specific receptor located on a neuron, it reduces the activity level of that neuron and makes it less likely to fire its action potential or release neurotransmitters. The two receptors of GABA are GABA A and GABA B. To focus on the ligand-gated GABA A receptor, GABA A receptors are ionotropic receptors that are the main target of many anxiolytic or hypnotic agents. When GABA binds to the GABA A receptor, it causes the opening of an associated ion channel that is permeable to the negatively charged ion chloride. When negative chloride ions flow into the neuron, they hyperpolarize the membrane potential of the neuron, and it makes it less likely for the neuron to fire an action potential. When negatively charged ions rush into the cell, this in turn increases the negative charge inside the postsynaptic cell, which makes it more difficult for an excitatory inputs to cause an action potential. Now, GABA A receptors are present in high concentration in the limbic system. GABA B receptors are metabotropic or G protein coupled receptors. When activated, they frequently cause the opening of potassium channels. These channels allow positively charged potassium ions to flow out of the neuron, again making the neuron hyperpolarized and less likely to fire an action potential. The actions of GABA are terminated by proteins called GABA transporters, which transport GABA from the synaptic cleft into neurons or glial cells, where it is degraded primarily by mitochondrial enzymes. Because GABA can reduce neural transmissions, increased GABA activity can have sedative effects. To be more in depth, this is a GABA chloride channel as a neurotransmitter being the GABA. The ion is called chloride. Chloride comes into the cell and it makes the inside the cell slightly more negative, and the effect of that is an inhibitory postsynaptic potential. This is a presynaptic neuron as well as a postsynaptic neuron and an action potential that comes down the cell. We also have a voltage-gated calcium channel. This allows calcium to come and go inside and cause fusions of these vesicles, in which these vesicles contain GABA. From the presynaptic neuron, these vesicles traverse across the synaptic cleft, and they will bind to a receptor on the postsynaptic membrane. The chloride ions then come into the postsynaptic neuron, causing an inhibitory postsynaptic potential. With rohypnol, it does not bind to the GABA recognition site located at the junction of the alpha subunits. Thereby, it doesn't induce the opening of the channel as directly as GABA. Instead, the binding of the rohypnol is a receptor site increase the affinity of the GABA for the GABA binding site, and this leads to increased frequency of channel opening in and turned into influx of chloride ions at the postsynaptic cell, which ultimately makes it difficult for the excitatory inputs to cause an action potential. So when the excitatory signals caused by fear and stress are inhibited, in this way the result is decreased anxiety and greater feelings of calm. In addition to that, rohypnol can also produce mild muscle relaxation as a result of their action on GABA. Receptors in the spinal cord or cerebellum and brainstem are prime examples of benzodiazepines, or rohypnol in that case, in which normally GABA inhibits the ability of neurons to send messages to neighboring neurons. In other words, it stops or slows the communication between neurons, although when rohypnol is in play, the drug enhances the effects of GABA, further increasing or decreasing, the communication between neurons. The decreased communication or depressant effect causes drowsiness and confusion and can even have a more serious effect such as sleep, coma, or even death. Thank you for listening.